This module is going to deal with the procedure and the steps which are required to perform the ddNTP sequencing procedure using Taq polymerase as an enzyme. पहला स्टेप है इस सारे प्रोसीजर को परफॉर्म करने के लिए, उसमें हमें जो टेम्पलेट है उसको और ओलिगो न्यूक्लियोटाइड प्राइमर को अनीलिंग की तरफ लेके जाना है. As you know that during replication or when we need to synthesize the new strand of the DNA using a template strand, then we need one primer that binds to the complementary region of the template strand, and it provides 3' hydroxyl group to the polymerase to start polymerization. So the first step would be annealing of the oligonucleotide primer or universal primer to the template strand. For that purpose, we are going to take single-stranded template DNA in 5 microliter volume and oligonucleotide primer 3 microliter volume along with 5x reaction buffer 2 microliter in an EP cup or microfuge tube of 0.5 milliliter volume. The second step is going to be the incubation. दूसरे स्टेप में जब हमने तीनों चीज़ों को मिक्स कर दिया, रिएक्शन बफर को, ओलिगोन्यूक्लियोटाइड प्राइमर को और टेम्पलेट को, अब हमें इसको इंक्यूबेट करना है 65 डिग्री सेंटीग्रेड पे. दैट विल फैसिलिटेट द अनीलिंग ऑफ द प्राइमर with the template DNA. By the time the incubation time is over, then we shift that tube to the ice and allow it to cool down for 3 to 5 minutes. अब इस 3 से 5 मिनट का टाइम जब हमारा सैंपल annealed with the primer cool down हो रहा है, we have to prepare the labeling mixture. For that purpose, we are going to take 10x labeling mixture, ddNTP, extension and termination mixture, radio labeled DATP, and these should be thawed before making a mixture on the ice. 3 से 5 मिनट में एक तरफ हमारे पास जो labeled the DNA template DNA is going to be cooled down, and we are going to thaw these reagents. Now we have to look at the number of reactions. We have how many annealing reactions, how many sample, and for each of the reaction we need to transfer 4 microliter of ddNTP termination extension mixture into color coded tube. Now look at this drawing. We have this tube that carries template DNA. Now the next step would be either we have 4 different tubes with different colors or with label like A reaction, T reaction, G reaction and C reaction, in which we are going to transfer 4 microliter of the ddNTP mixture, and this is going to be stored at ice because the volume is so little. If we don't maintain the temperature, then it is going to be evaporated because of the room temperature. Then we have to prepare the enzyme to be used for the sequencing reaction. Enzyme Taq polymerase is available in concentrated form. We have to dilute it using the dilution buffer. The ratio is going to be 1 to 7. That means 1 microliter or 1 part is going to be the enzyme and 7 part is going to be the dilution buffer. Now the concentration of this diluted enzyme is going to be 1 unit in 1 microliter. We need 2 microliter of this diluted enzyme for each set of 4 sequencing reactions. That means the final concentration is 1 unit per microliter, and this diluted enzyme is going to be stored on ice because if we are not going to maintain the temperature or storage temperature, then the enzyme can lose its activity and will fail to perform the polymerization reaction. In the annealing reaction that we prepared at step number 2, we are going to add 10x labeling mixture 2 microliter, radioactive DATP 0.5 microliter, DNA polymerase enzyme 8 microliters, and we are going to mix it and incubate it for 5 minutes at 45 degrees centigrade. This radio labeled DATP is basically to fix or to identify the starting point of the newly synthesized strand. After that particular time of incubation, 5 minutes, we are going to add 4 microliter of labeling buffer to the sides of each of the C, T and G tube. It is important here that we should not directly add this volume into existing volume in the tube. It should be added on the walls because it contains the radioactive material. So we are going to add on the wall, and then we are going to mix it by centrifugation at 2000 RPM. Those drops or those chemicals which we add in smaller volume on the walls of the tubes, when we are going to centrifuge it, they settle at the bottom and they are going to be mixed. The next step would be 5 minutes incubation at 72 degrees centigrade. After that particular time, we are going to stop the reactions by adding 4 microliter of formamide-loading buffer, and that is the last step for this particular procedure. The next, we have two options. Either we can store these 4 tubes at minus 20 degrees centigrade for further analysis later on, or we can proceed further to sequencing gel by using heat denaturation of these samples and performing the gel electrophoresis by using the sequencing gel, and we can read out the sequence of the DNA by the sequence or arrangement of the bands on that particular sequencing gel.